Being organized by the Department of Chemistry, Pandit Lalit Mohan Sharma, Government PG College, Rishikesh. Here is the agenda for today's webinar. During this pandemic era, the whole world is struggling with coronavirus disease, that is COVID-19, which is an infectious disease and is caused by a newly discovered coronavirus. Coronaviruses are a group of RNA viruses that cause diseases in mammals and birds. In humans, they cause respiratory tract infection that can range from mild to lethal. The virus that causes COVID-19 is mainly transmitted through droplets generated when an infected person coughs, sneezes, or exhales. These droplets are too heavy to hang in the air and quickly fall on the floors or surfaces. One can be infected by breathing in the virus if he or she is within the close proximity of someone who has COVID-19, or if he or she touches his or her eyes, nose, or mouth after touching a contaminated surface. This webinar emphasizes on the biology behind this virus and on the preventive measures, that is, mainly masking and the reason why one should mask oneself. So now I would like to invite the convener of this webinar and the head of the chemistry department, Dr. Hitendra Singh, for the formal welcome of all the guests and the participants of this webinar. Dr. Singh, please.
Thank you, Dr. Vibha. Good morning to one and all. It gives me an immense warmth and great pleasure to grace all of your presence in the interest of entire mankind. We are glad to have you all here to participate in the international webinar on the theme in Science Behind COVID-19 and its Prevention. That is going to be organized by the Department of Chemistry, Government PG College, Rishikesh. It gives me tremendous contentment to be presenting the welcome speech amongst the most esteemed personalities who have won accolades in their respective fields. It gives me a great pleasure to extend my warm welcome to our chief guest, Professor N.P. Maheshwari, the former Director, Higher Education, Uttarakhand, for accepting to become the chief guest of this international webinar. I welcome both the eminent speakers who have come here to share their vast knowledge and experience with all of us. I welcome the participants without whom it would not be possible to make this webinar a successful one. I welcome all my faculty members, my department members, faculty colleagues, and my technical staff for this organizing this webinar. The topic of today's webinar is Science Behind COVID-19 and its Prevention. Scientific knowledge of any theme helps us to aware about the mechanism behind that theme. And for this webinar, it would help us to understand the mechanism behind this pandemic and what would be our attitude towards the prevention of this pandemic. It helps an individual and a system also to set their goals, our work on that approach, and their beliefs or the faith in the potential system, and to improve awareness. Online seminars are organized and conducted for the general audience to gain insight and information that would remain effective in their daily life, exercises, and work culture. Our topic for discussion and debate for today's webinar was chosen keeping in mind the interest of each and every human being, those who are directly or indirectly linked with this pandemic. Some of them are corona warriors. So through this webinar, an individual is given a platform to identify their vulnerable spots, gain understanding about pandemic, focus on essential aspects of its prevention, improve their hygiene habits, maintain social distance, and provide practical communication skills with a social distance as well as the frequent changes that are needed to be in their lifestyle. So once again, I welcome you all to this webinar. And now I would like to hold this mic to Dr. Vibha to proceed this webinar further. Thank you. Ma'am, please unmute.
Is it okay now?
Yes.
So thank you, Dr. Singh, for a formal welcome to all the participants. Now I request the principal and the patron of this webinar, Professor Sudha Bharadwaj, to please come forward and share her kind words to all the participants. Principal ma'am.
A very good morning to you all. Chief Patron Dr. Kumkum Rautela ma'am, Director Higher Education Uttarakhand, Chief Guest Dr. N.P. Maheshwari, former Principal of this college and retired Director of Higher Education Uttarakhand. Speakers of today's webinar, Dr. Pinky Sharma, Principal Investigator, Wellcome Trust, DBT Fellow, Lab of Genetics and Neurobiology, Faridabad. Speaker Dr. Vishal Verma, Assistant Professor, Civil and Environmental Engineering, University of Illinois, USA. Convener Dr. Hitendra Singh, Associate Professor and Head Department of Chemistry. Organizing Secretary Dr. Vibha Kumar, Assistant Professor, Department of Chemistry, and all the participants. On behalf of Government PG College Rishikesh, I welcome you all in this international webinar on science behind COVID-19 and its prevention. Dr. Pinky Sharma will be talking about biology behind COVID-19 and Dr. Verma's talk on importance of masks in aerosol transmission. These two topics are very relevant today. I'm sure the talks are going to be very informative and will help everyone in understanding the COVID pandemic better. I again extend a very warm welcome to you all. Thank you.
Thank you, ma'am. As without your support, this webinar would not have been a truth today. Our chief patron and the Director of Higher Education, Dr. Kumkum Rotela, could not attend the webinar due to some important meeting, but she has sent her wishes for this program. Thank you so much, ma'am, for your wishes. It means a lot to us. Before I proceed, I just want to confirm, has Dr. Maheshwari joined? Vivek?
No मैम अभी तो कनेक्ट नहीं हुए सर.
अभी बात हुई थी तो he is about to connect.
Okay, ma'am. Let's wait, ma'am.
Can we wait for 2 minutes because he was saying he'll connect in 2 minutes.
Okay, ma'am.
Yes, Dr. Vibha. Professor Maheshwari is about to join us.
Okay.
They will take just one minute. Just 1 minute.
Okay.
There is some technical problem.
Sorry for the inconvenience to everybody. For 1 minute.
Professor Maheshwari is about to join us, and before he will join, I would like to share some information about Dr. Pinky and Dr. Vishal Verma. I think that both of our speakers worked in that field that is very relevant to this our today's topic, because COVID-19 is such a pandemic that affected the whole of the world within 6 to 7 months or 8 months from right now. Just a minute. Okay. Okay. And this pandemic era gave us such a lesson that how we can deal with any pandemic. For the, you can say that we have observed in 1920s. Okay, and for that we have prepared so much things, but right now we couldn't get any information from this from that pandemic that we have observed in 1920 or 1918. Okay, and Dr. Maheshwari is joining. So I want to give some technical assistance to Dr. Maheshwari. So Dr. Vibha will tell you rest of the things. Ma'am, please.
To confirm, has sir joined?
हाँ, अभी नहीं किया है, मैं उनको थोड़ा सा बता दूं. हाँ. उनको टेक्निकल प्रॉब्लम आ रही है. Is about to join us very soon. Ma'am, please you can take.
Actually, I'm waiting for him. I want him to join. Then I'll continue.
Okay.
Just wait for 1 more minute. I think he'll join in 1 minute. I'm again sorry for the inconvenience. Dr. Maheshwari has been the former director of higher education in Uttarakhand. He has also been the principal of our college. So when we joined in this college, he was a principal. And after that, he joined the directorship in higher education. And he has retired this year in February. He's a very gentleman, a very good man. What else can I say about him? That's just I want him to join, then I want to continue. Vivek?
Yes, ma'am.
Has he joined?
No, ma'am. Abhi to nahi, ma'am.
Just ask him if he's facing some problem, then please help him out.
Okay, ma'am.
He's facing some technical problem. I talked to him right now. Actually, he couldn't get connected with the password. So
Vivek, just call him and tell him the password on phone itself.
Okay, ma'am.
हेलो हाँ सर सर ज्वाइन कर लीजिए ओके सर सर बिकॉज वी ऑल आर वेटिंग फॉर यू क्योंकि ज्वाइन हो गया है सर सर चीफ गेस्ट को बोल रहे हैं ना अभी तो इसलिए आपका वेट कर रहे हैं तो कोई हाँ कर लीजिए सर ओके he is about to join within 1 or 2 minutes. Yes, Vivek, has he joined?
Dr. Vivek, continue, please.
Sir is joined?
Yes.
Okay. I'm once again sorry for the inconvenience and keep you waiting. So now we continue with the webinar because our chief guest has joined us. I find immense pleasure in introducing our honorable chief guest, Professor N.P. Maheshwari, former Director, Higher Education, Uttarakhand. He is a Doctor of Philosophy in Commerce. He has a long journey of 44 years in the field of higher education. During this journey, he had held many glorious positions, including principal of various UG and PG colleges, the Deputy Director of Higher Education, and also the Director of Higher Education. Many social organizations, including Rotary Club, has honored him for his outstanding social work. I want to take this opportunity to inform you that apart from all these academic achievements, he has earned priceless things in his life, that is dignity, honor, love, and respect. He is a man who is loved by all. We respect you, sir, from the depth of our heart. He has consistently proved his worth and have not failed in any of his endeavors. He has also started a fellowship for the students from his own pocket. I request our chief guest, Professor Maheshwari, the former Director, Higher Education, Uttarakhand, to shower his blessings upon us. Maheshwari, sir.
Sir, please unmute. Sir, unmute yourself. Unmute कीजिएगा.
First of all, I am very sorry. Due to electricity problem, I could not join the meeting within time. और इस इंटरनेशनल वेबिनार जो साइंस बिहाइंड कोविड-19 एंड इट्स प्रिवेंशन टॉपिक पर आयोजित किया जा रहा है, उसमें मुझे चीफ गेस्ट के तौर पर इनवाइट करने के लिए मैं आपका आभार व्यक्त करता हूं। श्री देव सुमन यूनिवर्सिटी कैंपस कॉलेज, गवर्नमेंट पीजी कॉलेज ऋषिकेश प्राय अपने कार्यक्रमों में मुझे शामिल करता रहा है और इसके लिए मैं पुनः आप सभी का महाविद्यालय के जितने भी प्रबंधन से जुड़े हुए लोग हैं उन सभी का आभार व्यक्त करता हूं। जहां तक आज के टॉपिक का प्रश्न है, साइंस बिहाइंड कोविड-19 एंड इट्स प्रिवेंशन, ये बहुत ही समसामयिक टॉपिक है और जैसा कि हम जानते हैं कि पूरा विश्व कोविड-19 की महामारी के दौर से गुजर रहा है। COVID-19 is the infectious disease caused by recently discovered coronavirus. This new virus and disease were unknown before the outbreak began in Wuhan, China in December 2019. According to medical scientists, the most common symptoms of COVID-19 are fever, tiredness, dry cough, and breathing problems. जहां तक ये प्रश्न है कि किस तरह से फैलता है ये कोविड-19, तो इससे पहले मैं आपको ये जानकारी देना चाहूंगा कि पेंडेमिक इन 2003 SARS सीवियर एक्यूट रेस्पिरेटरी सिंड्रोम हो चुका था और 2009 में H1N1 पेंडेमिक हुआ यानी कि स्वाइन फ्लू हुआ और 2014 में इबोला एपिडेमिक महामारी से पूरे विश्व में ये फैली और अब हम देख रहे हैं कि 2019 में कोविड की शुरुआत हुई और वास्तव में प्रत्येक वायरस के फैलने की जो दर है वो अलग अलग होती है और वैज्ञानिकों का मानना है कि एक वायरस का रिप्रोडक्शन रेट क्या है, ये बड़ा इंपॉर्टेंट इशू है। उदाहरण के तौर पर मीजल्स वायरस के फैलने की दर सर्वाधिक 15 to 18 है, अर्थात यदि एक व्यक्ति को मीजल्स होता है तो उससे 15 से 18 व्यक्ति इन्फेक्टेड हो सकते हैं। दूसरी ओर स्वाइन फ्लू से इन्फेक्ट होने वाले व्यक्तियों की दर सबसे कम 1.3 है, अर्थात एक व्यक्ति को स्वाइन फ्लू होने पर एक या अधिक व्यक्तियों को स्वाइन फ्लू से इन्फेक्शन होने की संभावना होगी। वैज्ञानिकों द्वारा कोविड-19 के संबंध में की गई रिसर्चेस से अब तक ये अनुमान लगाया गया है कि कोविड वायरस की इन्फेक्शन दर 2 to 3 है, अर्थात एक व्यक्ति को कोविड होने पर 2 से 3 व्यक्ति और इन्फेक्टेड हो सकते हैं। Many viruses replicate very quickly और जैसा हमें ज्ञात है कि कोविड व्यक्ति से दूसरे व्यक्ति के क्लोज कांटेक्ट में आने पर फैलता है अथवा यदि किसी व्यक्ति की इम्यूनिटी वीक है तो उसके जल्द इन्फेक्शस होने का भय रहता है। यही कारण है कि कोविड-19 एक ग्लोबल प्रॉब्लम बन चुका है। अब जहां तक इस महामारी से बचने का प्रश्न है तो बचाव का केवल एक ही रास्ता नजर आता है कि विश्व के समस्त देशों में जनता को वैक्सीनेट किया जाए, लेकिन विडंबना यह है कि अब तक इसका कोई कारगर वैक्सीन विश्व में उपलब्ध नहीं है। 9 अप्रैल के नेचर जर्नल के अनुसार विश्व में इस समय लगभग 78 वैक्सीन प्रोजेक्ट पर रिसर्च हो रही है और इस बीच खबर है कि ऑक्सफोर्ड यूनिवर्सिटी और यूएस बायोटेक्नोलॉजी कॉरपोरेशन के द्वारा वैक्सीन का ट्रायल किया जा रहा है। दूसरी ओर सोवियत रूस ने वैक्सीन बनाने का दावा किया है। तो इस तरह से वैक्सीन जब तक तैयार नहीं हो जाता तब तक हमें हमारे सामने केवल सावधानी रखकर या जानकारी प्राप्त करके ही एक मात्र बचाव का रास्ता है। और जब तक हालांकि इसमें भयभीत होने की आवश्यकता नहीं है और हम कुछ जानकारी रखकर और जरूरी सावधानियां रखकर कोरोना से बचाव कर सकते हैं, क्योंकि सावधानी ही बचाव है। और हम जैसा कि कहा जाता है कि इसके लिए हम लोगों को एसएमएस करना होगा। यहाँ एसएमएस से मेरा तात्पर्य यह है, एस से तात्पर्य है सोशल डिस्टेंसिंग, एम से तात्पर्य है मास्क और एस से मतलब है सैनिटाइजेशन। यानी कि हमको प्रमुख रूप से इन 3 चीजों पर ध्यान देना होगा कि हम दूरी बनाकर रखें आपस में, और मास्क का उपयोग करें और सैनिटाइजेशन का प्रयोग करें, अर्थात हम हाथ वगैरह बार बार धोएं साबुन से और सैनिटाइजेशन का प्रयोग करें। और साथ ही साथ हमें आंख, नाक और मुंह को न छुएं, ये भी जरूरी है। और साथ ही अगर कोई परेशानी पैदा होती है तो हमें डॉक्टर की सलाह लेनी चाहिए। और अंत में मैं केवल इतना कहूंगा कि जानकारी रखकर और सावधानी रखकर हम कोरोना से अपना बचाव कर सकते हैं। और जब तक वैक्सीन डेवलप नहीं हो जाता है, वैक्सीन मार्केट में नहीं आ जाता है, तो हमें केवल इन उपायों पर ही ध्यान देना होगा। और जैसा हम जानते हैं कि भारतीय संस्कृति में हम इन चीजों पर काफी जोर देते हैं। मास्क या सैनिटाइजेशन का उपयोग करने पर। और अंत में मैं केवल इतना कहना चाहूंगा कि लौट आएंगी खुशियां, अभी कुछ गमों का शोर है, जरा संभल कर रहो दोस्तों, ये इम्तिहानों का दौर है। और 2 लाइन और इसमें मैंने जोड़ी हैं कि कुछ सिखाकर ये दौर भी गुजर जाएगा, फिर एक बार हर इंसान मुस्कुराएगा, मासूम ना होना मेरे दोस्तों इस बुरे वक्त से, कल आज है और आज कल हो जाएगा, कल आज है और आज कल हो जाएगा। तो भयभीत होने की आवश्यकता नहीं है और हम सावधानी रखकर सोशल डिस्टेंसिंग के द्वारा और साथ ही साथ मास्क का उपयोग करके, सैनिटाइजेशन का प्रयोग करके हम कोविड से बच सकते हैं। और ये हमारे लिए एक तरह से अच्छा संकेत दिखाई दे रहा है अब कि जो मृत्यु दर है कोविड से मरने वालों की, वो लगातार घट रही है। और कोविड से जो जिनका इलाज हो रहा है इस बीमारी से बचाव के लिए, उसमें उत्तरोत्तर वृद्धि हो रही है, यानी कि ठीक होने वाले लोगों की दर लगातार बढ़ रही है। तो अंत में मैं डॉक्टर विभा कुमार का विशेष रूप से आभार व्यक्त करता हूं, जिन्होंने मुझे इस इंटरनेशनल वेबिनार में शामिल होने के लिए आमंत्रित किया और मुझे अवसर प्रदान किया। और साथ ही साथ इस इंटरनेशनल वेबिनार के जो कन्वीनर हैं डॉक्टर सिंह, उनका भी आभार व्यक्त करता हूं। और साथ ही साथ महाविद्यालय की प्राचार्य डॉक्टर सुधा भारद्वाज का भी विशेष रूप से आभार है कि वो प्राय अपने कार्यक्रमों में मुझे शामिल करती रहती हैं। इसी के साथ बहुत बहुत धन्यवाद। और सभी पार्टिसिपेंट्स इस वेबिनार में जो तकनीकी विशेषज्ञ आए हैं, उनके विचारों को सुनकर लाभान्वित होंगे, ऐसा मैं उम्मीद करता हूं, ऐसा मेरा विश्वास है। और पुनः आप सभी का बहुत बहुत धन्यवाद।
Thank you so much, sir, for the information you provided and for your blessings. Thank you, sir.
Welcome.
Now I would like to introduce our first presenter, Dr. Pinky Sharma. Dr. Sharma is the principal investigator at the Regional Center for Biotechnology, Faridabad, India. She is a recipient of prestigious Wellcome Trust DBT grant. She did her PhD from National Center for Biological Sciences, Tata Institute for Fundamental Research, Bangalore. She has done her first postdoc from Germany and another one from USA. She has published a number of peer-reviewed articles in highly ranked journals, including Neuron Nature. The focus of her lab is to understand taste and olfaction in invertebrates and its modulation using Drosophila melanogaster as a model system. So before I welcome Dr. Sharma, I would like to announce that you may send your questions at any time during the presentation. We will collect these and address them at the end of the presentation. And also a recorded version of this webinar will be available. So now I welcome Dr. Sharma to present her talk. Dr. Sharma.
Thank you, Dr. Maheshwari, for giving us already some piece of information and making my life easy. So that's really grateful. And I would like to thank Dr. Hitendra Singh, Dr. Vibha Kumar, and respected Principal Ma'am for arranging this webinar series and also inviting me to it. And hopefully the information we provide would be useful for everybody. I'm going to keep it a little simple so that even people who are from the non-biology background would be able to understand properly. And due to technical issues, I'm sorry I have to stop my videos because multiple devices are going on the internet connection. But I'll be happy to receive questions at the end, and I'll switch on my video again. Okay, so good morning, everyone. I would like to start my presentation now. As we all know that the world is currently witnessing a very severe health crisis of its time, and everyone is struggling to fight against this viral disease, which is called COVID-19. Almost everyone is at a grave risk in such an unpredictable scenario where range of symptoms are appearing in people and they are so variable from people to people, making this pandemic a real threat to the human race. Okay. Okay, so the novel coronavirus disease, it is basically the 5th documented pandemic after...
Sorry to interrupt, Dr. Pinky.
Yes?
We cannot see your presentation.
Okay. I cannot see that window where I was able to view everyone. So what should I do?
Just go to the presentation and forget about the window.
Go to the presentation. It's just that it is full screen. So I don't know how to. Let me see.
Just take your time. No worries.
You know, I was able to see myself earlier and all of you, but now that window. Okay, I'll just do escape and share the screen again.
Yeah.
Can you see me now?
No.
One second. So my presentation is there. It's just that
earlier we were able to see it, but now I don't know how come.
Yeah.
Just try it again.
Okay, just a
मैम आप ना अभी अपनी पीडीएफ और जो आपकी फाइल है मैम पहले आप वो ओपन कर लीजिए.
I have to just do the share screen again.
Yes,
so,
ma'am.
are you able to see something wrong?
Yes, ma'am.
Now is it okay?
Yeah. No, Pinky, it is okay.
Okay.
You can carry on.
So the novel human coronavirus disease COVID-19 is basically the 5th pandemic that has been documented, and it was reported in Wuhan, China, and later on it started spreading worldwide. So the coronavirus was officially named Severe Acute Respiratory Syndrome Corona 2 by the International Committee on Taxonomy of Viruses based on the phylogenetic analysis. And the spread that has happened is very, very fast. You can see the total number of cases here, which are like 3 crores, 85 lakh and 54,000, and total number of deaths are overwhelming, giving us a number of 10 lakhs plus. So it is one of the serious diseases. And right now, this is the scenario which gives us a total count of total number of cases and the deaths happening worldwide. And we are also on the top. So this is really a scary situation for India as well. So before I move on to coronaviruses, I would like to give a brief introduction on viruses in general. So basically viruses, you can see there are multiple viruses which are being shown here. Viruses are microscopic parasites and they are much more smaller than bacteria. They basically are not living when they are outside the host cells, but as soon as they enter into the host, or if they find a host, they reproduce very, very fast and they spread very, very fast to different part of the body. And based on the phenotypic characters, these viruses have their morphology, the kind of nucleic acid they have, either RNA or DNA, the mode of replication, and also the host organism and the type of diseases they spread. They have been categorized into 7 different classes, which are being shown here. So this is not the first time that we are witnessing a huge pandemic. There have been instances of major pandemics in the past, and coronavirus is one of them, and maybe one of the most deadly pandemic in the recent years. So basically all these pandemics have started because during the early civilization, when people were not moving that much, there were local pandemics, epidemics which stayed only in the local vicinity, and they were not spread to other countries or continents. But after the origin of like say big armies or movements of the human, whenever any pandemic has happened, it has moved to different parts of the world, making them a real burden on society and people. So you can clearly see the past pandemics here, ranging from smallpox to Black Death, plague, Spanish flu, SARS, AIDS, Ebola, and also now coronavirus disease caused by SARS again. So generally, based on what we have seen for viruses, coronavirus is one of the type of viruses which was invented in December 2019, Wuhan, China. And this is a spherical enveloped virus. You can see the structure of the virus here. It is around 120 nanometer in diameter, and it contains single-stranded RNA as a nucleic acid inside it. So what is strange and what is unique about this coronaviruses are the presence of these spikes on the surface, which are basically the culprits which are causing the harm to the human body. So these spikes are very unique, and coronaviruses cause a variety of diseases in mammals, birds, ranging from cow, pigs, and they cause mostly the upper respiratory diseases in chickens also. And later on, it all jump from the animal to the human, and then there's a spread of human to human. So I will come back to the structure and this morphology of coronavirus later on. So there are various number of human coronaviruses which are present, and they became 2 different lineages. And you can see that in most of the cases, their natural hosts are bats, and in some cases also it's rodents. And if we look at the SARS coronavirus 2 and MERS coronavirus, which belongs to coronavirus groups, they have caused severe acute respiratory disease in the recent past and now, but many of them also had mild symptoms which could be taken care very easily. So coming back to coronavirus COVID-19 again. So as I said, the coronavirus 2, it has a very unique and special structure, and these spikes makes them more unique. So these spikes outside, they basically come in contact with the membranes. Say, if they are coming through the nose or through the mouth or through the eyes, there are basically different receptors which are present where these coronaviruses can bind. So they recognize ACE2 receptors on various human cells. ACE2 receptors are basically present in most of the tissues and cells of the human body, and that's why it is making it a deadly disease to handle with. And once inside, so the attachment of the spike with the ACE2 takes the virus inside, and once inside, the virus can easily replicate with the help of some other peptides that help them to fuse their own membrane with the host membrane. They replicate, they make their multiple copies, they come out of the cell, and then they, from that particular cell or tissue, they start spreading to the other cell or tissues. So the replication cycle is very, very fast, and it's very unique. In the past, the other coronaviruses were not so severe because the kind of bonding that you are seeing here was not so tight. But due to the presence of these spikes, this is something which makes it so deadly that even the small number of viruses present on the human the cell surfaces of human cells can cause the disease. Basically, the severity will be different, but this is one of the specific concerns that has made this virus so deadly. So if we look at the different symptoms of COVID-19, people are experiencing fever, cough, shortness of breath, or difficulty in breathing. Sometimes they also feel chills, muscle pain, headache, sore throat. There is now the new symptoms are loss of taste and smell. There could be a congestion or a running nose and vomiting, nausea, and diarrhea. So all these symptoms are not necessarily to be present in all the patients, but if you feel that one of the symptoms are appearing in your body, and if you feel something different than usual, I think that is an alarming sign, and one should basically go and ask the doctor, if they have COVID or not. So one of the other thing is that not just that we should seek the doctor's consultation, but I think immediately we should not panic and we should take some protective measures, like washing your hands, as Dr. Maheshwari said that we should do SMS. So I think that's the best and the cheapest way to protect yourself and not to spread it further. And the other thing is that even if it is normal infection and not really COVID, one should just avoid the crowded places and try to, eat healthy and exercise daily. So there are different type of patients which have been recognized by the medical authorities, and each group are experiencing different kind of symptoms. So the first group is asymptomatic, and you might have heard this term many, many times. So the asymptomatic group basically are the dangerous carriers because they don't know that they have the infection and they're still moving outside. They're spreading the infection to everybody else, and they don't have very visible physical symptoms. The other class is where people are showing symptoms. So as I just discussed that what could be the probable symptoms if you catch COVID-19. So these patients basically should go to the doctor and seek some test and see if the severity of the symptom is just mild, or they need to be on ventilation or something. Then the third group is the largest group, at least so far that what we have seen. These patients actually develop flu-like symptoms. You will feel chills, you will feel fever, and also you can have breathlessness. So definitely you need to get admit and get the proper treatment. And the last group, where people are basically showing grave symptoms and pneumonia, are the ones where they have chest congestion, they cannot breathe, they have to be on the ventilators, and the proper treatment of 15 days should be taken in the hospitals. So if you feel, if you belong to any one of these categories and you are facing some kind of symptoms, I think it's important that we should not hide it and immediately go to the hospital or clinicians, get tested, and take the preventive measures and treatments to not spread it further and recover from it as soon as possible. So if we look at the timeline of COVID-19 infection, on day one, people can have, different kind of symptoms, like they can have probably fever, or they could have fatigue or muscle pain. They can have dry cough. They can feel some pain even in the throat. And it could also have diarrhea and nausea, which can persist for 2 days. By day 5, many people are facing breathing problems. If they are asymptomatic, maybe they will not face the same, but in most of the cases, they are having breathing problems. People on day 7 generally get admitted to the hospital, and then in severe cases, there are some distress in the lungs where the respiratory syndrome is happening, and they are not able to get enough oxygen and supply it to the other organs. And then on day 10, people can actually have worsening breathing problems. They are admitted to the ICUs. And finally, at day 12, the fever and other things could linger. If you are lucky, you will survive. But in many cases, we have seen that people, there are casualties and people are passing away. So we should not wait for day 12. From day 1, we should take proper care of our body and go to the doctor. The most important thing is that I know everybody is scared and they are taking precautions, but not all the infections that looks like coronavirus is actually coronavirus. You might be facing a seasonal cold or flu symptoms. So it is important that we should know what is the difference between coronavirus infection, seasonal flu, and common cold. So generally in coronavirus, it starts with the dry cough, which is generally not the case with the common cold. And then there is a fatigue or muscle ache on day 1 in coronavirus. It could have some other systems or breathing problems. There is never a case which showed that you have a running nose, which generally happens in the case of flu. So there are some symptoms which makes one disease different from the others. And I think if we go by this list of symptoms which are presented here, when one can easily identify what are you considering as coronavirus, what are you considering as flu and cold, and how to take the preventive steps. Okay, so these are the different tests which are present in the market right now. And this is generally how it works. So you might have heard all these terms again, but I think it's important that we should revise so that we are well aware of all these tools that are available for us. So the tests which are available, there are basically different kind of tests which are available in the market, but 3 different tests which are available right now are categorized under 2 categories. So the first category is diagnostic test, and the second category is antibody test. So the diagnostic test has 2 tests, where in the molecular test, these are basically these are basically done by using RT-PCR technique. So in this test, they will the doctor or the nurse will take the nasal or the throat swabs from the patient, and they will look for the nucleic acid or the genetic material of the virus in the patient nasal swabs. So they're looking for the RNA, and these tests are very, very sensitive compared to other tests, but these are very expensive and it takes very longer to get the result. In the antigen test, these are also called rapid test. These are easy to do, very quick, and the results can be out in 30 minutes. So what happens in these tests is the samples again are nasal or throat swabs. One would look for the presence of viral proteins in the swabs. You know, it could be a spike, it could be an envelope of the virus or the whole structure. So these are not based on the DNA or RNA material. That's why the detection is very, very specific. So these are called antigen tests, and antigen could be any protein, any glycoprotein which is present in the virus, and it's present in the nasal or the throat swabs. Then the second category of test is called antibody test. These are also called serological testing. So again, the serological tests are not that expensive. They are basically based on the results taken, assessed from your blood. But the problem is that people who are recovering or who has just started the infection, it is very difficult to detect viral load in those cases. So these tests are not very sensitive. So sometimes if the viral infection is there, but the antibodies are not produced in a huge number, the antibody test will not be able to recognize the level of antibodies and the presence of coronavirus in the blood. So based on the specificity and the accuracy, molecular tests based on RT-PCR are the best, but antigen test also gives 95% specificity and accuracy. So one can go for this first and later they can confirm with the use of RT-PCR test. And although we don't really know how the virus has emerged, how the outbreak of COVID-19 has started, but as Dr. Maheshwari also said that it started from Wuhan, China. So the suspicion is that the virus started from from bats, where there was a cross-species transmission, probably pangolins, and then or it could be some other animals. After a zoonotic transmission, it went into the human beings, and then from there, the human to humans transmission started. And then people who were traveling with the infection, they went to different countries. There were some local transmission, when they were not maintaining proper hygiene, when they were not washing their hands and not taking care of their health, the virus spread to all over the world. And since it is 2:3 ratio, people were not aware of it and they were kind of ignoring it in the beginning. Now the result is that it's very, very quickly spreading everywhere. So we don't know if there is a risk of viral spread from the animals to people. And what I mean by this is that we already know that bats are the culprits. The virus might have evolved from the bat and then from bat to pangolin and then to human beings. But there is a very little data which suggests that there are other animals as well who could spread the infection to other people. So there was a report from New York Zoo which has found only one tiger which was found to be positive for COVID-19. But it has also been seen that mice, chickens, pigs, and ducks, they do not have the receptor where the virus can bind. So these animals will not be able to spread. In India, we see chicks, pigs, mice everywhere. So we don't have to panic because we know that the virus won't infect them. But on the other hand, dogs have some receptors present in them. So there's a chance that dog can mildly affect people, but it's not going to be so severe. since the numbers are very, very small, we still don't have enough data that could suggest what could be the potential carrier, in the animal kingdom that could further spread to different humans. So one would wonder, if the virus is so deadly and it is causing such a harm to humans, how come the bats outsmarted the virus for 65 million years? So the virus present in the bats actually not really causing any disease to the bats, but it is causing millions of deaths of human beings otherwise. So what recently researchers have done, they did the genomic sequencing of bats, and then they took 42 categories of mammalian species, and they compared the gene sequences between bats and all the other mammalian species. So what they have found, that there are at least 10 genes that bats have disabled, which mount inflammatory responses against infection. So there was some modification in the genes of inflammatory response in the bats, and that's why, if the virus is present in the bat, will not cause any harm to the bat. The other reason is that they also have extra copies and modification of many antiviral genes that explains their high tolerance to coronavirus. So I think the bat has evolved in such a way that now, no matter how many coronaviruses are going to infect bat, they will never get any severe disease within their own population, but they can easily spread to other animals or to humans. So who is at the higher risk of receiving these symptoms? So far, whatever reports are there, it suggests that people who are in an older age, which are basically 85 years old and plus, people with lung problems, because it's a respiratory disease. So already people having lung disease or respiratory problems are also at the danger risk. People with heart disease, diabetes, obesity, cancer, and some other blood disorder, or weak immune system, or if they have kidney or liver diseases, can also easily catch the infection. So, all these diseases we are listing here because these are the organs where ACE2 receptor is present in abundance. If you remember how the virus goes into the body, so these are the organs where the ACE2 receptor is present in abundance, and these are very easy targets for virus to spread there. So one has to be very, very careful if one is suffering from such diseases. And recently it has been shown that there is a big connection between obesity and COVID-19 connection, and one should not ignore this. If one is overweight and if they have BMI of 40 and above, those people are actually at higher risk. So it has been shown that basically in obese people, the obesity prolongs the duration of virus shedding. So basically, if you are fat or obese, the virus load will always be very high because they grow on the adipose or fat tissues, and also from there they go to the lungs. So individuals which have rich source of adipose tissue, they have increased number of ACE2 expressing cells, and then virus can easily attach to these cells there. Also, the immune system of obese people is weak, and also their lung capacity is weak. So one has to be very, very careful, and one should try to maintain a stable body weight if in case you catch viral infection. One of the other things that has been recently found is the cytokine storm in patients. So you might have heard that people are recovering, but all of a sudden, they passed away because what is happening that when the treatment is given, the immune system is producing lots of proteins as a defense mechanism, but they are produced in a manner which is much more than the expected. So these are called cytokines, and the cytokine load is so much in the body that the body is not able to handle it. So because of this also people are dying. So it is important that, we should not wait for the severe infection to occur. We should take the precaution from the very first beginning. So generally, cytokine cells are the kind of cells that move from one place to another place, and they are generally generated in response to a immune response in the body. One of the other symptoms that have been listed as, the potential markers for COVID-19 is the loss of taste and smell. So basically, even in asymptomatic patient, when they have no other symptoms, they can also feel that suddenly they are not able to smell, suddenly they are not able to taste anything. And this is alarming because generally it doesn't happen. So when you have cold, stuffy nose, or you have ulcers in the mouth, you can feel the same. But if you are healthy and all of a sudden you feel that there is a loss of smell and taste, that is the first sign that tells you that you have a COVID infection. And there are very simple tests that you can do at home. You can take a piece of garlic and try to eat something, that you appreciate a lot, like something that gives you a good flavor, and you can ask yourself, is I'm smelling and tasting properly? So these are the little tests that you can do at home and self diagnose yourself. And I think one should do it on a very regular basis, because we all are going outside, we go to the market, we are working, and sometimes we forget to wear masks, so one can easily catch infection. I think this is the test that can tell you if you are still all right or if you have already caught the COVID-19 infection. Okay, the other thing is that it has been also reported by many groups that men are more vulnerable to the infection than women. So the differences between men and women is that first is the presence of different kind of hormones in the body of women and men. And generally, women, uh, the kind of immune system that women have, it is much more effective in handling the viruses. So progesterone and estrogens and progesterone are the hormones which are present in humans, and they are very easily able to protect against the virus. But testosterone, which is present in men, it is not handling the virus that well. So that why these differences are occurring, it is not known scientifically yet, but these could be the potential differences that are telling why men are getting more infection compared to the women. And also the genetic makeup, if one's immune system and body type is very different, that can easily catch the infection between men and women. That could also explain that maybe genetically men are more prone to catch such type of infections. And also it has been found that ACE2 expression is greater in men compared to women. I really don't know the reason for this, because we all have the same organs in the body, but it has been found that ACE2 is expressed in higher number in men. Okay. So if we look at the current scenario, the number of cases increasing and the number of deaths are happening, I think it is getting very, very difficult for the medical authorities to, take this load on them and also treat the patient. So now people are using artificial intelligence to handle all this. So definitely, one has to treat to basically uh, tell the machine what is its job. But after feeding so much data, artificial intelligence is actually helping doctors and other researchers in handling the pandemic. For example, like the proper screening, tracking, and predicting the current and future patients can be done using the artificial intelligence. There are various kind of detections which are and diagnose which has happened with the help of artificial intelligence for the coronavirus. and to develop the drugs and vaccines and to reduce the workload for healthcare workers, artificial intelligence has actually proved very, it's like a blessing in disguise right now. So people are working on to see how artificial intelligence can be used even for other pandemics or the future pandemics, so that whatever is happening right now, that can be handled in a very short span of time, and we don't really have to wait such a long to understand the disease so much. So we all know that we all have to wear masks, but I think it is important the kind of mask that we should wear, and also, how much distance one should maintain, even if you are wearing a mask. So there are different kind of masks which are available in the market. You can see if you are not wearing a mask, the droplets from your mouth can easily spread to a longer range. If you wear one layer mask or two layer mask, the risk is reduced. But if you use surgical masks, which are properly made, the risk is very less, and the kind of droplets that come out are very few. They are almost none. The similar scenario is there when you are coughing. So when you cough, the droplets actually go very far. One cannot see the droplets, but they are there in the air. And coronavirus infection is airborne. So it is very important that we control this behavior by wearing the mask. The other thing is that sneezing makes it even more worse, because when you are sneezing, the droplets are even going further. So the take home message from this slide is that one should wear a mask, and probably we should wear mask which has multiple layers. And you can see here how when people are talking to each other face to face, how much distance we should maintain and how masks could be effective in spreading the disease. And you don't have to really buy the mask from home. There are multiple ways, and one can stitch that home even with your hands, and it's very easy just to even just tie up a piece of cloth around your mouth and protect yourself and others by maintaining a proper distance. So these pictures are from the past pandemics, and you can see that they look very much similar what we are experiencing right now. Nothing has changed between the pandemics that has happened in the past and the current pandemic. You can see people are wearing masks. The social distancing were maintained when people were admitted to the hospital, and the severity caused by pandemics in the past is no less. Even the kids, they had to wear masks and they had to stay home to protect themselves. But what did we learn from the past pandemics? The first lesson that we have learned is that yes, the social distancing works. Yes, wearing mask is important and it works to stop the spread of the disease. So we should not be shy of wearing mask, or if even if we are meeting our dear ones and our family members, I think it is very important that we take care of ourselves and others. The other thing is that we should not forget that even the younger ones, they can easily catch the infection, although it has been said that kids are not really contagious and they are not showing any infection, but that's not true, because kids might not be showing infection, but they're spreading it to many people. So it is important that even the young ones are taken care properly. We do not blame the people who are sick right now, because they are already sick. The important lesson is that we should protect ourselves and we should not make ourselves sick. The other thing is that yes, we have to wait for the vaccine, and vaccine will work and the pandemic will end. So I know that people are waiting for this, and they are waiting for a magic to happen, but we all have to be patient to see how much we can control it by ourselves until we get the proper vaccine. So there are right now people, I think I see a lot of people wearing masks here, and it has been shown even in the past, even in the past pandemics, And wearing mask is not new. It has been started long back when the Spanish flu, when the industrialization happened in Japan. So Japan is the first country who started making masks, and people were forced to wear masks so that they can protect themselves from the viral or bacterial diseases, and also from the strong dust that comes from the industrialization. As an example here, this is a very old poster from one of the Japanese ad, and you can see here the person who is not wearing the mask is spreading the viral load to the other people. So one should not make this mistake. Even if you feel healthy and even if you don't have the infection, please do wear mask. So right now, it has been shown that the virus is mutating very, very fast. There are different, so these color codes are showing different strains of virus which are floating in different countries of the world. The good news is that yes, the virus is mutating, but the kind of mutations that are happening in the virus are not so serious. So the changes that are happening in the genome are not causing even more serious disease. There is no structural change. So there is a hope that whatever vaccine that will be produced soon will be effective against all the evolving COVID strains. And even now, you might have heard this that Trump was recently infected with the virus. So even the people who were not believing that COVID is actually so serious and we need a vaccine and all, even they are suggesting that yes, one should have a vaccine, but we need to undergo proper trials, and it has to face the trials in such a way that whenever kids or anybody, whoever is getting vaccinated, they are not at the risk. So one has to wait for the proper vaccine to come out after the trials are done, and I think it's not going to happen so soon, before end of 21 or early 22. So this is the new term which has been floating recently, and people are thinking that in the absence of vaccination, herd immunity could be one of the best policies to protect people. But as a biologist, I would not suggest that we should go for herd immunity, because people are not able to understand what exactly herd immunity is. So people are thinking that if you have infection in most of the population of the country, then everybody else will get some kind of immunity and they will not carry the infection further, and everybody else will be immune to it. But that's not correct. For the herd immunity, one needs to get vaccinated. So what is shown in here is that if one person is infected and very few are vaccinated, that one person is enough to spread the infection to many people. All right. But if many people are vaccinated and one person is infected, then most of the people who are vaccinated and who are not vaccinated are immune to the virus or the bacterial disease. So we need to understand this concept very clearly, so that we should know that for getting the herd immunity, one needs a vaccination, and without vaccination, when one cannot achieve herd immunity. And this is very risky if one applies herd immunity, because in the absence of vaccine, people who have already pre-existing diseases and people who are older in ages, they will be the first one to die. So we cannot let our people die just because some people don't understand what herd immunity is, and our socio-political scenario doesn't understand, and wants to apply this in the system. The government has launched Arogya Setu app, and I also have it in my phone. We all are using it at home. I think it's an important app, and one should install it in your phone, because generally we don't know how many people are affected in our close vicinity. But if you have the app, at least your 500-meter range or your close proximity, you will see how many people are infected. And by doing this, you can actually protect and guess if you are in the course of getting the infection. So I think this is an important tool, and having this in your phones will make your life little easy. The other thing is that boosting your immune system is very, very important, because people with the compromised immune system are the ones who are catching the infection. So eat proper meals and balanced diet, which has zinc, iron, and multiple vitamins in that. We should stay physically active. We should sleep enough. If you are stressed, if you are not sleeping enough, and also you have the infection, you might not be saved from the disease later on. One should drink enough water and stay calm and do not panic. The other thing is that mental health is a stigma in India. I think people should talk to each other and discuss if they have the disease, and they can actually go and seek some help from their friends or relatives or people in the family. And one should make sure that, if somebody is having the infection, we should not isolate them. All right, that we cannot meet them physically, but there's always a way to talk to them and give them some hope, so that the disease is not progressing further, and they can take it positively and handle it positively. Above all, I always feel that no matter what people are facing, money crunch, and people are losing jobs. So this is the right time to show that humanity is above money, and we should definitely try to each help each other in whatever way we can do. We should develop hobbies, do exercises, and try to find our ways to get engaged. If you're working from home, if your kids are not going to the school, this will help to keep a positive attitude. In one of my last slides, I would say we should not forget our soldiers who are working day and night and risking their own lives. Around 600 doctors have already died, which is a big number worldwide. I think we should appreciate their help. They are not given salaries. We should try to do whatever best we can do for them. And with the last slide, I would like to end my presentation here and say that there are different means, how the pandemics end, and definitely it will end one day. So one type of end is that people are no more catching the infection, there is a vaccination, and no more deaths are happening. The other way is that people are not at all having the fear of the pandemic anymore, and right now we are in that state. So basically we know that the pandemic is around, the infection is around us, but we are not worried, and we are taking it very positively. We are doing our day-to-day activities. So with that hope, I hope that soon we all be able to resume our work. Kids will be able to go to school. And, as Dr. Maheshwari said, that Aaj is, the Kal will become Aaj and Aaj will become Kal. This routine will keep going. So one should always be positive and optimistic of the current situation and handle it in a very positive way. Thank you so much for your attention, and I would be very happy to take questions if you have any.
Thank you, Dr. Sharma. Your talk was wonderful and full of it was very knowledgeable and full of many facts which many people didn't knew. So this is very true that the movement in or out, in other words, globalization, is the major reason for the spread of any disease throughout the world. The session is open for the questions. If anyone of you has any question, you can come up with it. So I have some questions from the participants. Someone is asking that after having symptoms, can one distinguish that he or she is having simple flu or coronavirus disease without going through a test?
Yes, so if you remember one of my slides where I have shown the differences between coronavirus, flu, and common cold. So one can identify those differences. Plus, I think it is very, very important that you do a test for your smell and taste. You can also do a 10-second test by holding your breath, and you can see if you feel uneasy after, you are not breathing for 10 seconds. And if there is any distress caused in the lungs or in your body, I think that is an alarming sign which can tell you that you might be suffering from corona.
Okay. One more question we have. Here, the participant is asking that the fear of virus is more dangerous than the virus. How can one overcome this fear?
Yes. So I think it's a very nice question, and that's why I discussed a little bit on the mental health, because if you are in a panic state or a fear state continuously, you're stressing your immune system and you are making yourself more vulnerable to catch the infection. So don't do that. Even if you catch the infection, it is all right, because there are preventive measures, there are precautions that one can take, there are treatments available, and it is working. It's not that it is not working. So one should not fear that much. And if you catch it, I think do the right thing, go to the doctor first.
Okay. Now, another question related to the Arogya Setu app. The people are asking that how reliable is the app when the reports are not very reliable these days?
Okay, so I think the app is reliable only if we are being honest with ourselves. If you are feeding your information correctly to the app, then it is helping people to identify how many around us are infected already and which way you should avoid. So it depends on the transparency and the information which are provided by the people who are suffering. The other thing is that do the testing from the centers which are authorized, who have the staff, like research centers are also doing the testing for COVID patient. Don't go to anybody. And if you feel that the first time it is negative and you still feel that you might be having corona, I would say go for RT-PCR test, because they are more reliable and more specific and sensitive test.
Okay, thank you so much. We are getting many comments from the participants. Like they really liked your lecture. They are saying it was a very wonderful presentation and very useful for them. So
Thank you so much. Yeah, thank you so much.
So once again, thank you, Dr. Sharma. So your talk was really wonderful. Okay. Now I would like to introduce our second presenter, Dr. Vishal Verma. Dr. Verma is an assistant professor at the University of Illinois, Urbana-Champaign, and his current work is focused on assessing the health impacts of ambient air pollutants. In his 13 years of research career, he has published more than 40 peer-reviewed articles in highly ranked journals and has presented his work in at least 50 seminars, meetings, and conferences, including several invited talks. He is the past chair of the Health-Related Aerosol Working Group at American Association for Aerosol Research. He is the guest editor for the journal Atmosphere and has earned numerous awards and recognition for his work, including the National Science Foundation Early Career Award. I welcome Dr. Verma to please enlighten us with his informative talk. Dr. Verma.
Thank you, Vibha, for the nice introduction. And thank you for inviting me for this wonderful platform to present some of the work or the information I have gathered so far in this area. So is my slides visible to everyone?
Yes.
Okay. So the title of my talk is the role of aerosols and the mask in the transmission of COVID-19. So at the beginning of this forum, I think Vibha said about something about the transmission mode of this COVID-19. I think I would like to differ something here, and it could be somewhat, contradictory to what has been, mentioned over the WHO website also, because I think one missing point in this whole game is that the role of aerosols has not been recognized, which I would like to actually highlight here. So, I mean, it can actually could be a game changer because the aerosols do not behave and they do not follow the law of 6 feet. So, okay. So this is an overview of my talk that I would basically talk about the transmission modes of this COVID-19, and then the effect and importance of the size of the aerosols in the transmission, and then what evidence we have for the transmission of this virus through aerosol mode, and specifically what interventions we can actually take to control this aerosol transmission of COVID-19. So before I actually talk about the transmission mode, I think I would like to make this distinction between the droplets and aerosols. And honestly, this is, I mean, I found it quite complicated, before I actually started looking into this area. I'm an aerosol scientist. I do not have any biological background. I'm not a doctor, but what we have been taught in the aerosol science and engineering is that aerosols are the particles suspended in the air. But this definition is somewhat vague because for how long they are suspended in the air is not clear. For example, a 0.1 micrometer particle can remain suspended in the air for hours, but for a 100 micrometer particle actually will remain suspended only for seconds. So in the aerosol community, the adopted definition for the aerosol particles is that any particles less than 100 micrometer in size is called the aerosol. But surprisingly, I recently found that this definition does not match with the medical community. In the medical community, the particles above 5 micrometer are called the droplets, and the particles below 5 micrometer are called the droplet nuclei or aerosol. So I think this, probably this distinction in the definition of aerosol versus droplet was existing, but it was not actually noted before the beginning of this pandemic. And this actually caused a lot of confusion in this guideline about 6 feet, as I will show that if the transmission of this disease is occurring through aerosol, they do not follow the law of 6 feet. So let's stick to the aerosol community definition that in the following slides, I will say if the particles below 100 micrometer in size, I will call them as aerosols, while the particles above 100 micrometer, I will call them as droplets. Okay, so what are the transmission modes? So this slide, I think, summarizes these different transmission mode. One mode we all know about, that is through the contact, that, a patient, he or she might touch his nose or, the hands or anything is infected with the virus, and then it can touch the other person and then transmit it. Or that infected person actually can touch a surface, for example, the cell phone, or let's say if I put these hands on the table and infected this table with the virus, and someone else actually came and then touched the virus, touched this table, and then, so this is called the fomite transmission. And then with this infected hands, if someone touches eyes, nose, that's another, that's the same kind of contact mode. The other mode could be this spray borne, which is basically, someone sneezes or cough, and then these big droplets, which I like, the particles above 100 micrometer are directly inhaled by the other person. So they are directly hitting it. So remember, this is airborne, but this is direct hitting. It's not aerosol because the particles are above 100 micrometer. And then this third mode, which is probably kind of neglected in the medical community, is through the aerosol mode, that particles below 100 micrometer actually, so when someone sneezes, it's impossible that he or she will just release these big droplets. it's like almost inevitable that there's a spectrum of the particles. Some would be big, some would be small, and this small aerosol, I mean these small particles called aerosol actually can change the picture, like because uh, they come in different sizes, and based on their sizes, actually they can deposit in various places in our lungs. For example, the particles around 100 micrometer, they can deposit in the upper respiratory system, like just the nose, and the particles between 10 to 15 micrometer, they can actually just deposit in the tracheal region, and respiratory aerosol can actually go much deeper into the lungs. Um, so this is a comparison, like how big a particle is, when we call about this 100 micrometer, 5 micrometer. So the diameter of the human hair is around 60 micrometer, and the this quarter of the circle is shown here as the diameter of the human hair. Then a PM10 particle, meaning a particle less than 10 micrometer in size, will look like something, will look something like this. And a PM2.5 particle, that means the particles less than 2.5 micrometer, will look like this small, ball in this big yellow circle. Okay, so this is, I was talking about the size of the aerosol. What is the size of the virus? So influenza virus is around 0.1 micrometer in size, which is around 100 nanometer. And this SARS-CoV-2 virus is 120 nanometer or 0.12 micrometer. And the other respiratory viruses like rhinovirus, adenovirus are around the same size, like, so rhinovirus is 30 nanometer in size, 0.03 micrometer, and adenovirus, which is another type of respiratory virus, is actually around, again, 100 nanometer or 0.1 micrometer. So we see that the size of the virus is very small, like 0.1 micrometer. So does it mean that it will spread only through the aerosol? Not necessarily, because when someone sneezes or coughs, this virus is not naked, meaning it just doesn't come out by itself. It is always in this big particle where this virus is sitting here, which is a size around 0.1 micrometer, but it's in this respiratory fluid, which can actually go up to like 0.5 micrometer, or can be, I mean, like it can actually go to about around 100 micrometer. So why I'm talking about this size? The size of the aerosol or the droplet, whatever we call, is very important, because the size will determine how long it will stay in the air, how far it actually can travel, how quickly it will go, it will fall on the ground once emitted, and where, like, once we inhale this aerosol, in which part of your respiratory system it will deposit. Why this is important? There have been studies showing that if the aerosol actually reaches deep down in your respiratory system, it has a higher chance of developing the infection than the aerosol depositing in the upper respiratory region. So the same virus sitting on a smaller particle has a higher chance of developing the infection than the virus sitting on the bigger particle. And finally, how efficiently it actually can be removed by the mask we are wearing. Okay, so let's actually take the first question: how long it will stay in the air? Now, the people who have been working in the aerosol community, they probably are aware of this equation called the Stokes law, which is given by this which basically tells you the settling velocity of the particle as, in terms of the this equation, which basically gives the size of the particle, the density of the particle, slip correction factor, and the dynamic viscosity of the particle. So if we know all these parameters, then we can easily calculate the settling velocity of the particle. Now, if we know the settling velocity, then what we need to determine how long it will stay in the air is the distance. And so basically we divide this distance by the velocity, and we can actually get the time, right? So if you assume that the particle is emitted from the nose, which is around this, like, height of 1.5 meter, then basically, you just need to divide this velocity by the distance, 1.5 meter, and you can calculate the time. And this is, I'm showing actually here, that this is the time for an aerosol to settle from a height of 1.5 meter, depending upon its size. What information does this plot give actually? So it shows that an aerosol of 10 micrometer in size, it can remain suspended in the air only for 5 minutes. But as its size decreases to 5 micrometer, it can actually remain suspended in the air for 40 minutes. So basically, a decrease in the size of the aerosol by half increases the residence time in the air by 8 times. Okay, so this is information. This is useful information we should keep in mind, because it will be used in our further assessment. Okay, now this plot actually shows, so we know how much time aerosol can remain suspended in the air. That is in the still air. Now, if I mean, in the room or so, like, we don't have a kind of a completely still air. We always have some sort of air velocity. Even if we assume the indoor air velocity is like very low, around 5 centimeter per second, then this aerosol actually will go somewhat in the horizontal distance. So for example, this 5 micrometer aerosol actually can, with the indoor air velocity of 5 centimeter per second, it can easily go with that time we showed to around 100 meter in distance. While the 30 micrometer aerosol, will not be able to go further, like it will just drop there. So this is, why this is important, because if the transmission is actually occurring through the aerosol, it clearly shows that they are not following the law of the 6 feet, which we are hearing from the WHO or the CDC. And in the upcoming slide, I will show basically there are evidences which are showing that this aerosol transmission of the virus is important. Okay, but another question is, does the size of the droplet or the aerosol we have been talking about, does it remain constant? So you know that the virus is not naked. It actually comes in this droplet. And most of the content of that droplet is like water, so it's volatile. Only the non-volatile content is only 1 to 9%, So what does it mean? So let me just show you the typical content of a respiratory droplet or the aerosol. So this is excluding the water. So basically the composition of the non-volatile fraction of the droplet. So like protein accounts for like 24 to 89%. And if you talk about the virus, it's actually almost negligible. So it's not zero, but it's like 0.001%, something like that. And this is the electron microscopic picture of that droplet. So what controls this size? Like once we cough or once we sneeze, how does the size actually vary? So what parameter which is going to control that? So in the indoor environment, the only parameter which actually controls the size is the relative humidity. And how we can actually use this relative humidity to know how the size of the droplet or of the aerosol once emitted is going to change. For that, we use this equation. So the people working in the aerosol community, this equation is very familiar to them. It's called the Kohler equation. And this basically tells you, if we change the relative humidity of the room from 100% to 20%, then how does this shrinkage of the particle happens? Like how the particles varies from a given size to a smaller size. So this is the ratio of the final to initial diameter. So this could look like a little complicated, but I mean, we can use this information to calculate that if a droplet of 100 micrometer, which basically contains here, this coronavirus and this salt and protein particles, and if the size of this droplet of the aerosol is 10 micrometer at 100% relative humidity, if the relative humidity changes from 100 to 20%, it will evaporate. So the water will go away. The virus will still be there, and the protein and the other components like salt will still be there, but it will shrink from 10 micrometer to 4 micrometer. Why is this important? You just saw that a 10 micrometer particle remains suspended in the air for few minutes, but 4 micrometer particle remains suspended in the air for 40 minutes. And in 40 minutes, if there is a significant air velocity in the room, it can easily travel to like maybe 50 meters. So the 6 feet, guidelines is probably not applicable here if the transmission is occurring through the aerosol. This is a recent study actually published. It's on the CDC website. That okay, we know that the aerosol can travel, that far, but does the virus remain alive for that much time, period of time? Like, we talked about 40 minutes. So this study showed through this, the plaque assay they actually conducted on the impingement samples, which have been collected, from the aerosol suspension. They found that the aerosol remained alive for up to 16 hours in the air. That means even if the aerosol is actually moving and traveling around a distance of 100 meters, the virus will stay alive. Okay, so this was all about the mechanism of the mechanistic insights on how far these particles can go. But what evidence do we really have to show this transmission of the SARS-CoV-2 in the aerosol mode? So recently there have been many studies. I think the first study I am aware of is that the study conducted in the Wuhan hospital itself, like, from where this pandemic started. They measured the particle size distribution of this inactive SARS-CoV-2 virus. Why I am saying inactive? Because the methodology they used, that is not able to keep the virus alive. They basically collect the particles. Actually, they use the impactors, which have been developed by my PhD advisor, Kostas Siotas. They use the Siotas impactor to actually collect these particles of different sizes. Now, the virus doesn't remain alive in that methodology, but they just measured the RNA, and they found that there was significant presence of this virus in the particle size less than 1 micron. Like, you can see even in 0.1 micron. So in 3 different hospital zones. Okay, this was inactive virus. Because there was no there is no very developed methodology or the robust methodology to collect the infectious virus. But there exists one biosampler which actually can collect the live virus, because the way it collects the particles, it actually collects the particles directly into a solution, which keeps the virus alive. And that study actually collected, I mean, there have been not a lot of studies, but this study actually was conducted in Singapore in this airborne infection isolation room at the National Center for Infectious Disease in Singapore. And they used a biosampler in different sizes through which they can actually collect the aerosol of different sizes. And they found the presence of this live infectious virus in the size range of 1 to 4 micrometer. So that means, yes, the virus, even the infectious virus, which is able to continue the infection, actually exists in this size range of 1 to 4 micrometer. And 4 micrometer aerosol means it can actually travel, it can remain suspended in the air for 40 minutes, it can travel like 50 meters, which is way more than the 6 feet. So this study, published in August 2020. This is very interesting study because it showed that this virus actually is not only emitted only in the during sneezing or coughing, but also during the breathing in the initial stages. So I think this study basically shows, I mean, it gives the hint that why the asymptomatic people who have not been even coughing or sneezing, how come they are able to, transmit this virus? Because even, I mean, they found that this virus is released during the breathing also. And during breathing, earlier, I mean, other studies have shown that the particle size distribution is generally towards the lower sizes, which can easily transmit very far. And recently there have been super spreader events. One study I'm aware of is that in South Korea, in which I think on the 11th floor, people were sitting, maintaining somewhat social distancing, but out of 1100 of people working in that building, 96 cases were there for the positive for the coronavirus. And one study I know is actually occurred in this Washington, in this Skagit Valley Corral, in which people were, kind of rehearsing. And the interesting thing about both of these studies is that people were actually taking the precautions of, the usual precautions of hygienes, like, washing hands, maintaining social distance, but they were not wearing the mask. And I think the common features of the super spreader events is that they are generally crowded. People were talking a lot, and there was insufficient ventilation, and people were not using masks. But they were maintaining social distance, and they were also washing their hands a lot. So the only mode of transmission which can actually explain this super spreader event is that the transmission was occurring through the aerosol, because only aerosol don't obey the law of the 6 feet. And I'm going to show you that we can control that by wearing the mask. So aerosol transmission seems to be the only dominant route to explain such a super spreader outbreak. Okay, what can we do if the transmission is occurring a lot through the aerosol? So our intervention strategy should stem from our understanding of the science of aerosol transmission. So we know from where the aerosols are coming, we can control them at the source. If there is aerosol, concentration filling up a space, we can dilute it. And finally, we can actually filter them through the air. So I will talk about these different interventions. So the first intervention I'm talking about is that, this using the mask. And this study I'm referring was published in Science, which basically showed that how the universal masking is really helpful in controlling this the transmission of this disease. For example, this infected asymptomatic person is just speaking, maybe not even coughing or sneezing, but it basically gives you a burst of the aerosol. And if you are in the proximity of that person and not wearing the mask, you are at the maximum exposure. If you are wearing the mask, then you will be somewhat protected. But the best thing would be that that person also wears the mask. So, which actually clearly shows that it, which I think Dr. Pinky Sharma also showed in her slide, that, how this masking actually can substantially reduce the release of this aerosol as well as the droplets. I will talk briefly about these different types of masks. For example, this is the surgical mask. Now, this mask is intended to keep the wearer from spraying droplets onto the others. Now, there is something called the N95 respirator. This is basically to protect the wearers for the exposure of the inhaled particles. And why is it called the N95? Basically, is that this mask is able to remove more than 95% of the most penetrating particles. When I say most penetrating particles, it's not the smaller size particles. I will talk about it in a minute. It's around the size of around 300 nanometer. So this is something, I would like to show here is that what are the different mechanisms by which this mask remove the particle. So one common misconception about the filtration of the particles in filters or the mask is that it probably removes the particles by sieving. That's not true. It removes the particles by by 4 of these different mechanisms, which are called the inertial impaction, gravitational settling, diffusion, and interception. And I will talk about it briefly. So what is inertial impaction? Inertial impaction is that a heavy particle is, moving smoothly, but then there is a sudden change in the direction of its streamline. And why does the streamline changes? Because, near the fiber, obviously the streamline will change, fiber of the mask or something. So now this heavy aerosol is so heavy that it's like a heavy person is running fast, and then you suddenly ask that person to change the direction. He or she will not be able to change. He will just drop there, fall there. So it happens with the particle. A big particle can easily is not able to follow the direction. It will just stop there and stick to the fiber. So that is the inertial impaction. Diffusion is that, the very small particles, they don't go in the straight line. They always go like this, like zzz, like, they always vibrate, So because they don't go in the straight line, they by chance, so if they started from this streamline, they will end up in this streamline, which is actually very close to the fiber, and they will eventually hit this fiber and then get removed. So this is called the diffusion. And then gravitational settling is that particle is too heavy, it just falls down onto a fiber. And finally, interception is also called the chance meeting, which is basically the particle actually was in the streamline which was going to hit to the fiber. So it just intercepted on the fiber. So interception is what you can call as the sieving action, which is not a dominant mechanism in the filters or the mask. So I just talked about it, that the filters or the mask don't collect by sieving action, but by these 4 different mechanisms I just talked about. So this is the testing facility we actually developed in our aerosol lab at UIUC for testing the filtration efficiencies of different masks and respirators. So this is basically, we are following the NIOSH protocol, which is this National Institute of Occupational Health and Safety. So we basically generate these sodium chloride particles of a known particle size distribution using this atomizer, and then pass them through a diffusion dryer, which is then neutralized by this neutralizer. And then, these aerosol are then actually fed to this stabilization chamber, because the concentration is very high, so we have to dilute it, and then we can basically measure the particle size distribution through this scanning mobility particle sizer, and then pass them through this this filter holder. So what we do is basically we cut a small section of the mask and put it into this filter holder, and we measure the particle number concentration using this condensation particle counter, both upstream and downstream of this filter holder, to know how much this mask placed into this filter holder is removing, is being removed. Okay, so we just talked about the different mechanisms by which this mask or the filters can remove the particles. So what can we expect a typical filtration efficiency curve to look like? We know that the small particles have a mechanism for the removal, which is diffusion, right? The big particles also have a mechanism of removal, which is this inertial impaction. So they both are removed very efficiently. It's the middle size range of the particle for which we do not have a dominant mechanism. And as you can actually, sense from my, from my description, this is the size which is most difficult to be removed by this mask, which is between 100 to around 500 nanometer, because we don't have a dominant mechanism for the removal of these particles. So when we say N95, we basically look at the particles of around this size range. So an N95 particles should be able to remove more than 95% of this particle, which is around 300 nanometer in size. So this is kind of counterintuitive. Like, we would expect, okay, the smaller particle will be easily, will pass through, bigger particles will be captured. No, it's actually the middle size particle which are not efficiently removed in the mask. and N95 works perfectly in that sense. Okay, this is so, this N95 masks have been in dire shortage during this pandemic. So we also developed a method in our laboratory to actually, to reuse this mask. And what we did is a very simple thing, because we know that this dry heat is able to inactivate the virus. But does it, I mean, in addition to inactivating the virus, is it able to retain the filtration efficiency of this mask? That's what we tested actually. So we found that it is able to decontaminate the mask, but it also is able to retain its filtration efficiency, and also the fit test, because the mask has to be really properly fit onto your face. Otherwise, a lot of air actually can easily pass through the gaps around your nose and mouth, so it will not serve your purpose. And what we found is that this, by using this dry heat, using a simple, this electric cooker at your home, you just put that mask into this electric cooker for like 50 minutes, and your mask is ready to reuse again. And we tested this with, not with the coronavirus, but with other viruses which are also very infectious, and we found that they were like, it's actually able to provide a very high decontamination efficiency for this mask. At the same time, this mask, even after this decontamination cycle, like heating cycle of up to 20 cycles, they were able to keep their filtration efficiency. Like, so they were N95 masks, meaning they were supposed to be removing more than 95% of the particles, and they were still able to maintain, like, so even after 20 cycle, the filtration efficiency was like, above 96%. And we also measure the pressure drop. Why pressure drop is important? Because if the pressure drop is very high, you will feel it. I mean, you will feel it in the sense that, you will not be comfortable, because it will actually create a lot of resistance in the air. So you will not be comfortable wearing that mask. So we found there was no pressure, there was no change in the pressure drop of this mask, even after the heating cycle of up to 20 cycles. So that means there is basically no change, no deterioration in the filtration, in the performance of the N95 mask. So we just learned, okay, that in the filtration efficiency, that how is it a function of the particle size? The same thing happens in our respiratory also, like in our respiratory system also. Because our respiratory system, starting from our nose to the lungs, is actually a kind of filter. And basically these four major mechanisms govern the deposition of these particles in our lungs. Like, again, those four functions for these four mechanisms, like inertial impaction, gravitational settling, diffusion, and interception. So, and in fact, our this viscous mucus layer in our air walls actually make sure that, the particles, once deposited there, it's not re-entrained. So it's even more efficient. So what you see here in the this human respiratory system, in the upper respiratory system, you see a lot of bands, right? What does band mean? The band means that there's a lot of change in the directions, right? So what will happen to the bigger particles? They are going to deposit in this upper part, right? In the trachea, there is no clear mechanism, right? Because it's a straight pipe. But when you go actually to this bronchial region, the flow which we are inhaling through the nose is going to be divided, right, into multiple branches. And when the flow is divided, basically what mechanism becomes dominant? The diffusion, right? Because that means the aerosol, which is actually, not going into the straight line, is going like, hanging like this, it actually has a higher chance of getting deposited in this lower respiratory system. So basically, the same these processes we learned are going to govern this deposition of the aerosols in the lungs, which basically tells you that in the pulmonary system, which is the deeper part of the lungs, is going to be smaller particles deposition, right? So the small particles are going to deposit in the lower respiratory region. The upper respiratory region, which is the nasal part, this actually going to be dominated by the bigger particles. And then in the bronchial region, because there is no clear mechanism, because it's kind of a trachea, which is a straight line, there is not much significant deposition. So it basically tells you that why it is important. we just talked about that a virus sitting on a smaller particle has a much higher chance of going much deeper into the lungs. And other studies have shown from the medical community that if the infection starts from the lower respiratory region, it actually has a, it actually shows much more severity than the infection is starting in the upper respiratory system. So interventions, the physical distancing, it's important to control this ballistic or the droplet infection. Like for example, if this person is actually talking, so if you just increase your distance from that person, you don't know whether it's asymptomatic or symptomatic, but you basically are able to reduce your exposure to this high cloud of the aerosol concentration, right? And if you wear the mask, that's even better. Another way of reducing this aerosol concentration is the dilution we talked about, right? And this is actually important in the indoor environment. So like you have a high concentration of the aerosol, what you can do is just dilute it. How you can dilute it? Open up your windows, put a fan so that this concentration of the high concentration of the aerosol is just vented out. And then if you have a negative pressure in your home, then eventually the fresh air actually will come from the cracks or or whatever openings you might be having in your homes. So but basically try to remove that aerosol concentration building up in your home to outside. This is particularly very important in the United States, where we have a central HVAC system in which all the air, I don't know about I think it's important for the buildings, for the school buildings, for the occupational buildings, that where most of this air is actually passed through a duct, and then it's conveyed through a duct, and then it is passed through a filter. Now that filter has to be really efficient to remove these aerosol particles, and as we talked about that, so these HVAC filters, heating, ventilation, and air conditioning filters, are available in different MERV ratings, like so they start from MERV 0 to MERV 16, and we know that a MERV rating of 16 is equivalent to an N95 mask. So if you actually use a high efficiency HVAC filter, it will be able to reduce the aerosol concentration, because what is it actually doing is basically pulling your inside room air and then passing it through a filter. But if that filter is weak, then it will keep on recirculating that same air with a high concentration, so the aerosol concentration will keep on accumulating. So in every cycle you can actually reduce that concentration. Other thing you can do is in an indoor environment is putting an air purifier or the air cleaner, which is a very simple device. What it does is that it basically pulls the air around it, pass it through a filter which is inside this purifier, and then push this clean air through this air purifier. And if you don't have the access to this air purifier, a simple thing you can do is you can make your own purifier. So if you have a box fan, you can just buy this HVAC filter, which should be available in the market, and fit it over it. So it's basically the same thing as the air purifier. It will pull the air but pass it through this filter, and then push this clean air through it. So in fact, there have been many variations of this box fan filter. So finally, I would say that avoid these 3 C's, which is crowded places, because we know that in the crowded places, the aerosol concentration is going to build up. Close contact settings, confined and enclosed spaces. So these avoid these 3 C's, and definitely wear a mask if you have to, you cannot avoid these 3 C's. And this is my last slide, that this SARS-CoV-2 virus is transmitted through both direct contact and indirect contact. Please don't ignore this indirect transmission through the aerosols, and poorly ventilated display places promotes this transmission largely through the aerosols. Physical distancing and other measures hygienes do have an important impact on controlling this transmission. Thank you.
विभा अनम्यूट कीजिए अपने को, डॉक्टर विभा.
Okay. Thank you, Dr. Verma. So the session is open for the questions. Anyone can come up with their questions. So Dr. Vishal, I already have some questions for you from the participants. The
Sure.
first one is, does the acidic content of air affect the aerosol? Acidic content of air.
Can the I mean, that person like elaborate that question? What do you mean by the acidic content? Like in
What
the indoor
I
air?
think is, he's a student. He's asking, like,
I
if
think he want to know about the air quality.
yeah.
I mean, as far as I know, is that in the indoor air is not acidic, because there is no acidic aerosol present in the indoor environment. So the indoor air is not acidic. If you're in the outside air, and you're wearing mask, then you don't have to be worried about this virus, because the concentration of the virus is going to be too dilute. The aerosol mode of transmission becomes really important in the indoor environment. So and I think his second question is also that, could UV rays affect the aerosol?
Yeah.
Yes, it definitely affects. So I do not work on UV, but I know a lot of researchers have worked on this, that, in fact, what they suggest is that while you actually are replacing your HVAC filter, you can also put a UV lamp, so that it actually not only filter the particle, but at the same time, if you keep on filtering the particle and keep on collecting it over the filter, over time, that becomes infectious, right? I mean, your HVAC filter itself. So when someone is going to change that filter, that person is at a high risk of getting the virus. So you should probably actually put a UV lamp there also. So yes, that UV is very efficient in killing the virus.
Okay, there are some more questions. A few days back, the government was announcing that N95 mask is not very efficient in removing the virus, but a triple layered mask is useful. So how true is it?
It's not true.
It's not true.
It's impossible. Yeah. And if it is really an N95 mask, so the question could be that, is it really an N95 mask? Because, and as per the original definition of N95 mask given by NIOSH, which is National Institute of Occupational Safety and Health, that it is able to remove more than 95% of the particles. No mask can be more efficient than that. A 3-layer mask we have tested in our lab, they are efficient up to an extent of 70 to 80%, depending upon the layers of the mask.
And the last question for you: can the mask be used after washing with detergent or only after autoclaving, that is heating?
Okay, that's a good question. So do not wash your N95 mask, because there's another mechanism of removal specifically on the N95 mask, which is this electrostatic charge. So they basically use a special type of fiber. If you put that into your washing machine or so, that electrical charge will be removed and its efficiency will highly degrade. For the surgical mask, I'm not sure, but I will suggest even that mask, do not wash it, because it seems to me that they also use some, like, this the surgical masks which is manufactured by these different companies, they use some sort of charging on their fibers.
Hi, Vibha. I have a question. Can I ask it?
Yes, you're welcome, Pinky.
Vishal, thank you for a nice presentation. I just want to add something on the mask and also ask you a question. So one thing is that, different surfaces are holding the virus for a different duration. So like metals can keep the virus maybe a little longer compared to the clothes or plastic or other surfaces, right? So I think it is.
No, I think I know about the study which showed that the plastic, I think plastic hold the virus for up to 3 days, and then metals like stainless steel actually hold it for a day, and the copper, actually the survival rate of the virus on copper is only a couple of hours. So
yeah, gold and copper are the best, I think.
Yeah.
Yeah. So, if we look at the fabric that is basically in use currently, can we not opt for options like silk or something where basically the virus particle is or the droplet is not absorbed inside? Like the mask can be multi-layered, but the outer layer is such that we can prevent the virus coming into, whatever route, like the nose or the mouth part.
Yeah, so I'm not sure, like, if there has been a study on studying the survival rate of the virus on the cloth. I mean, I think I'm aware of one study which actually studied on cardboard, metals, and um, on this plastic. But I think that could be an interesting idea, that we actually see that. I think what you are trying to say is that if you use is a cloth which
Yeah,
doesn't
so no
allow.
mask is 100% proof, completely giving immunity and completely blocking the virus particle, and especially the mask with the valve outside. I think they are the most risky ones because you are
Yes.
able to breathe through them, and the virus
Yeah.
is just 120 nanometer. It is very, very small. So do not use
Right.
this mask.
I 100% agree with you, Pinky, on that. This mask with the vent, they are actually dangerous to the person in your neighborhood, because when you exhale or something, they actually release this cloud of aerosol.
Yeah.
So any more questions from anybody? All the participants are welcome to ask their question. So I think there are no more questions. So a wonderful presentation, Dr. Vishal.
Thank you.
I would like to thank you for enlightening us with such a wonderful talk.
Thank you, Vibha, for inviting me.
Thank you.
Okay.
So you all will be getting the feedback form on your mails, and after filling it, you will receive the certificate. So we are heading towards the end of the webinar. And here I would like to invite the convener of the webinar, Dr. Hitendra Singh, to come forward and present his vote of thanks. Dr. Hitendra Singh.
Yes, thank you, Dr. Vibha. It is extremely an honor and privilege to propose the vote of thanks on this occasion to our esteemed guest. After thanking the Almighty God for making today's occasion a success after some technical uh, starting outbreaks, I would like to thank our chief guest, Professor N.P. Maheshwari, former Director, Higher Education, Uttarakhand, who had found some time to grace this occasion. Thank you very much, sir, for your presence here. Then I would like to thank Dr. Pinky Sharma and Dr. Vishal Verma, our invited speakers, who despite of their busy schedule has found some time to enlighten this program with their expertise knowledge. Dr. Pinky covered all the aspects and made the topic very relevant and understandable to each and every one, those who are having some doubts about this pandemic or COVID-19, and especially through this attractive presentation. I like that presentation very well, very much. And thank you, Dr. Pinky, so nicely presented this typical COVID-19 pandemic.
Thank you, Andrew. Thank you.
Thank you. And I also thank Dr. Vishal that covered the presentation so nicely that till now it was not included in our hygiene practices, how to get escape from the mouth droplets, and how we can get prevented through the droplets or aerosol transmission from one way to other, and most importantly, why. That is most important thing, why we should avoid these droplets and how to get escape from these types of droplets. So both presentations were very nice. I thank you our invited speakers once again. Next, I would like to thank principal and patron of this webinar, Professor Sudha Bhardwaj, for her guidance and encouragement in all our efforts. And I would like to thank two other members of our advisory committee for their constant advice and logistic support, especially Dr. Virender Singh and Dr. Sachin Sharma, who made sincere efforts to arrange this meeting practically feasible. So thank you, Dr. Virender and Dr. Sachin. I cannot forget to thank all my participants who has some spare this time to make this event a successful one. I would like to thank each and every member of the department for encouraging us to arrange this webinar on the current topic of COVID-19, as two of our members are the survivors from this pandemic. Dr. Shakunj and one of our lab assistant,
Dr. Seema.
Dr. Seema, and one of our lab attendant, that is Prashant. May God bless them long life. The last but not the least, I would like to thank our technical supporter, Mr. Vivek Rajbhar, who did a fabulous job to make such a nice arrangement for all of us. So thank you, Vivek. And after that, at last, I would like to thank Dr. Viva for organizing such a beautiful webinar and arranging all the invited speakers for such a beautiful webinar here. So thank you, Dr. Viva. Now, I would like to share my view with Dr. Viva that he worked very hard for this webinar and arranged all the logistic supports on his own. So thank you once again. Now it's your turn.
Thank you, Dr. Singh. Thank you so much. At this time, our principal and the patron of this webinar is busy in some meeting, so she is not available here. So I would request you, Dr. Hiten sir, again to declare this webinar closed.
जहाँ तक मेरी बात है, मैं तो इस वेबिनार को क्लोज करने का सोच नहीं सकता. अभी मेरे सामने डॉक्टर प्रोफेसर महेश्वरी सर हैं, तो मैं हमारे फॉर्मर डायरेक्टर से परमिशन चाहूंगा कि सर कैन वी क्लोज दिस वेबिनार.
Sure.